Hi everyone, welcome back. We ended our last lecture going through some of the major synapomorphies or the 6 synapomorphies for the land plant clade. And I ended with this slide where the land plants, which we also call the embryophytes, are those that have the young sporophyte, that is, or the embryo that's supported by the gametophyte. What I would like to do now is to move into the different land plant groups and actually start to look at the major innovations along the evolution of land plants, both non-vascular and vascular plants, and look at how and where those major innovations evolved. And so our first group that we're going to look at is the bryophytes. Bryophytes contains 3 main groups: the liverworts, the hornworts, and the mosses. And mosses are ones that people are most familiar with. These, just as a reminder, the bryophytes are non-vascular plants. So these are plants without vascular tissue. And as a result, these have to have a small size. And they typically, because they can't move water a long distance through vascular tissue, and they typically live in wet places as a result of this as well. We'll talk a bit more about their biology that restricts them to these wet areas in just a little bit. So these are non-vascular plants. When we think about the life cycle, we talked about life cycles and the alternation of generations in the last lecture. When we think about the life cycle of these are gametophyte dominant. What that means is that all of the green tissue, the body of the organism in these liverworts or in the hornworts or in the mosses, all the green tissue, that is all gametophyte tissue. So the haploid part of the life cycle actually dominates in bryophytes. When we look at, as a result, the sporophyte is actually dependent on the gametophyte. So these are gametophyte dominant and sporophyte dependent. And so when we think about the sporophytes on these, the sporophytes of liverworts are actually embedded in the top of this gametophyte tissue. This long stalk on the hornwort, this is the sporophyte. This is the sporophyte portion. And then in mosses, the sporophyte is this stalk and capsule part of that moss. So the non-green parts in that case are the sporophytes. And in all of these, the sporophyte is dependent on that gametophyte generation. So it's dependent on it as an embryo when it starts out and it gets that energy transfer, like we talked about through the placenta on this last slide. But it's dependent throughout the life of the sporophyte. And I'll show you a nice detailed life cycle of the bryophytes in just a little bit to go through that. But bryophytes, these 3 groups, they have these morphological similarities in that they're non-vascular, they're small size, they're gametophyte dominant and sporophyte dependent. And so these were always lumped into a group together. When I sketched out our phylogeny of these before, we had our Paraphyletic green algae that was sister to land plants. And the land plants, we had our non-vascular bryophytes, and we had our vascular plants. And it turns out that this is actually not correct, this part of it specifically. And what's not correct about this is that bryophytes are paraphyletic. And so these don't represent one lineage, but actually 3 independent lineages. Whoops, I can't spell. 3 independent lineages of the tree of life. I'll show you what that looks like in a second. And so, as you might imagine, just like with the green algae, when we saw that charophytes and chlorophytes were actually not monophyletic, it changes the interpretation of some major characteristics and major morphological traits that we thought were due to a common ancestor of this monophyletic group. It turns out that's not the case. So bryophytes, gametophyte dominant, sporophyte dependent, not monophyletic, they're paraphyletic, and they represent these three separate lineages. And so here we have our green algae, our paraphyletic green algae. And so you'll notice that whenever a group is paraphyletic, I put that name, the common name for it, in parentheses, or sorry, in quotation marks. So green algae are paraphyletic, bryophytes are paraphyletic, and bryophytes are paraphyletic in that they have these three independent lineages. It turns out that liverworts, so these is our common ancestor of all land plants down here. Liverworts are that first branch off of the land plant phylogeny, followed by hornworts and then mosses. And as we can see, certain characteristics then, the evolution of characteristics changes. And so one of those that I want to emphasize right now are stomata. I'll show you what stomata are. But the reason this one is interesting is because in that bryophyte group, hornworts and mosses both have stomata, but liverworts do not. And so that was always a trait that was difficult to understand until we were able to resolve the phylogeny and figure out that, oh, these are not monophyletic. Liverworts diverge first, and then hornworts, and then mosses. And so that helps us explain the presence of stomata. I'll also talk about these other two hydroids, as well as an upright gametophyte that we see in mosses, but we don't see in liverworts or hornworts. Okay, so let's take a look at some of those characters, starting with the stomata. And so stomata and pores are basically holes in the plant body. And why do we need those? You remember that we have the cuticle as one of our land plant synapomorphies. And that cuticle is that waxy layer that seals off the plant body and prevents water loss. Well, you know that for photosynthesis to occur, we have to actually bring carbon dioxide into the plant body. And so while the cuticle prevents water loss, it actually stops gas exchange as well. Well, that's a big problem. And so stomata and pores are both necessary to allow for gas exchange, and specifically to allow CO2 in. For photosynthesis to occur. And so if we take a look at the cross section of this leaf here that I showed you before for the cuticle, on the underside of the leaf, we actually have a couple of openings on that lower epidermis. And so stoma is a singular for stomata. Stoma is singular, stomata is plural. And so we have these openings in the plant body that actually allow for gas exchange to occur. But remember, I said that liverworts don't have these, but then mosses and hornworts do. And so how do we reconcile that? How do liverworts actually go ahead and have gas exchange occur? Well, liverworts have pores. And so what pores are just simple openings in the plant body. And so here's a liverwort shown down here. This is what we call a thalloid liverwort. So the body of the liverwort is actually pretty flat, like a piece of paper, a little thicker than a piece of paper, but it's flat bodied. And on the surface of those, if we zoom into the surface of these, we actually this thallus liverwort, we see these little pores. And so this is a pore here. That's the pore itself. This is an actual opening. If we look at that with an electron micrograph, we can see a little bit more structure, and we can see that opening there. And then looking at a cross section of it is the best. We see the opening where gas exchange can occur. But we also see that this is surrounded by this air chamber, and this then helps limit water loss. So we have this opening in the plant body where gas exchange can occur, but to make it so that all the water doesn't just evaporate out, we have this sort of humid boundary layer that's inside of that air pocket in there. And that humid boundary layer makes it so that you don't lose water too quickly. But in liverworts, we just have these simple pores. So pores allow for gas exchange in this liverwort lineage. So just those have it. But it allows for gas exchange. Because of that, though, liverworts are limited to very moist environments. So where do you find these? If I were going to go out and look for liverworts right now, I would head out to looking along the edges of ponds and creeks and some permanent water where it doesn't dry up. This is where you're going to find liverworts. And if you go out into the rainforest on the Olympic Peninsula or into the rainforest that we find up in our cedar groves, we actually, much of what people call mosses that are hanging off of the trees in those forests or growing all along the trees in those forests are actually liverworts, not mosses, a mix of mosses and liverworts. Anyway, so liverworts have just pores, well, but hornworts and mosses have stomata. And how are stomata different than these simple pores in the plant body? Well, they have a bit of a different structure and a bit of a different physiology in terms of the way that they work. And so hornworts and mosses with their stomata, and all of vascular plants with their stomata. So remember, stomata evolve here. That means everything in this monophyletic group, hornworts, mosses, and vascular plants all have stomata. And they all work very similar to each other. And so on either side of the pore or the opening, so if we look at the stomata in this hornwort here, we have a pore. And then on either side of that, there are two cells. And those two cells are called the guard cells. And they guard against water loss. And so when it's dry out, these can actually be closed down. The guard cells can close that pore down. And when the air is relatively humid or the water is quite available, those guard cells can be opened up. We won't lose too much water. And so they can actually, both hornworts and angiosperms, as you see here, have stomata that are very similar to each other with those two guard cells and that pore making up those openings in the leaves. And so it's important they can actually open and close these dependent on availability of water. So can open and close based on water availability. That's an important difference between that and a simple pore. Okay, how do they do this though? And they actually do this the same mechanism, and they use what's called a potassium pump to do that. And so one of the first thing that happens if a stomata is closed, then the plant needs to open that stomatum, potassium is pumped into the guard cell. As a result of pumping potassium into those guard cells and increasing the ion concentration inside of the guard cell, the ion concentration inside the guard cell becomes more than the concentration outside of the guard cell. And so because of that, water then can, by osmosis, diffuse into the water, into the guard cell. When water goes into the guard cell, what happens, that cell becomes turgid. Turgid is just a word for like when you blow up a balloon. The balloon's flaccid to start with. You blow up that balloon, and now it's turgid. So turgid is turgid is when it's all pumped up. So that guard cell gets pumped up, and when that guard cell gets pumped up, what happens is this pore opens up. So you pump up the guard cell, and you open that pore in the middle, and so the stoma opens. And then to close this, potassium is pumped out. Oops, phone ringing. So potassium pumped out of the guard cells. And when potassium is pumped out of the guard cells, the water then goes back out of that guard cell, and the cells become flaccid, guard cells become flaccid. So H2O out, flaccid cell, and the stoma closes. And so that's the way that the stomata, so the plant can actually regulate when it's opening and closing its stomata. Okay, hopefully that makes good sense. And so this is a big innovation, because now this allows these lineages, hornworts, mosses, and all the vascular plants to actually move out of the wettest environments and start to live in drier environments. Okay, so let's talk about what are some of the problems that we have on land. And so there are a number of problems, right? When we move on to land. And the first one of those is desiccation. And so how have land plants dealt with desiccation as one of these major problems? Well, we've talked about two of those right now. The first is the cuticle. And then right along with the cuticle, because this then limits actually gas exchange as well as preventing water loss, we've got pores and stomata as important solutions to that problem of moving out of an aquatic environment and onto land. Another one, which I mentioned briefly, but I'll emphasize here, is sporopollenin. Pollenin, that's right, covering the spores. So you remember sporopollenin was a desiccation-resistant polymer that actually surrounds the spores in land plants. This is one of the things that allows the spores to be preserved in the fossil record. It also helps with desiccation on land. So this is an important characteristic there as well. When I talked about sporopollenin, I also talked about long wavelength UV radiation. And so as you move out of the water and onto the land, UV radiation becomes a big problem. Sporopollenin helps with that too. Sporopollenin, I see I didn't spell that right on the top one. It should be like that. So sporopollenin also protects those spores against UV radiation. Another one that I hadn't mentioned here is polyphenolic compounds. And so there's a whole bunch of biochemistry that plants do that allow them that protect them against UV radiation. One of the main groups of polyphenolics that are found in land plants are flavonoids. And flavonoids absorb UV radiation. So that's protection against that UV radiation. Another problem that we have on land is that oxygen, actually, when you have those pores open in stomata, oxygen actually inhibits the fixation of carbon dioxide by the enzyme system that's used in photosynthesis. So in photosynthesis, there's an enzyme called rubisco. Rubisco is actually responsible for fixing carbon dioxide out of the atmosphere into a usable form by the plants, but oxygen actually inhibits that Well, there was a land plant synapomorphy that I mentioned before that actually solves this problem and that, or ameliorates this problem, and that's the glycolate oxidase enzyme system. And so what this does, it allows rubisco to fix carbon dioxide with oxygen present. Okay, the last one is nutrient acquisition. When you're in an aqueous or an aquatic environment, you are absorbing your nutrients through your body. So the body itself, through osmosis, you can actually bring a lot of nutrients in passively. But once you move onto land, you've lost that ability, and so you're actually not able to do that anymore. And so there's some evidence both from molecular phylogenetic data as well as from fossil evidence that fungi and land plants arose around the same time. And it's hypothesized, this is a really cool area of study, if people are interested in this, that there's a symbiotic relationship, or the symbiotic relationship between plants and fungi facilitated the movement onto land. And that's pretty cool. I want to emphasize that this is a hypothesis. This is a hypothesis that phylogenetic and comparative analysis studies, as well as experiments, we could get at, and so this would be a cool one for people to pursue. Okay, so I bring all of this. I bring these problems up just to sort of emphasize how our these different characteristics and these different traits in land plants actually are solving these problems. So the structure and function kind of relationship that we have when we move from that green algal ancestor to moving out onto land. And it turns out that charophytes, remember charophytes are sister to the land plants, are actually pre-adapted in many ways, or in several ways, for this movement onto land. And so a couple of those, I just want to mention a couple of those things that we think are pre-adaptations. One of those is that charophytes are freshwater and near shore. And so it's not a far stretch for them to make that movement onto land. And these near shore environments, you constantly have sort of fluctuating water levels, and these are variously exposed at different times. And so that movement, the evolution of these characteristics to deal with that exposure makes good sense. Charophytes also have polyphenolic compounds, remember, like those flavonoids. For UV absorption. So that's cool. That's something that's shared with that land plant group. Remember that charophytes also have spores with sporopollenin. And so that is another pre-adaptation for the movement onto land. And then the last one, which is a cool one, is that if you remember the life cycle of the Charophycean green algae, they're primarily haploid life cycle, so haploid dominant life cycle. And then they have one small diploid phase, and that's the zygote. So the zygote itself is diploid. So once fertilization between sperm and egg happens, you end up with a diploid zygote. That's the only diploid cell in the life cycle of the Charophycean green algae. And importantly, those are actually retained on the mother, on the haploid body. And so that zygote is retained on the haploid body. And that haploid generation supporting that diploid zygote is one step away from the embryo, which is when, if that zygote were to divide into a diploid embryo, like we see in embryophytes, the land plants, one step away from that embryo actually, from that embryo being retained and the embryophyte characteristic or synapomorphy of the embryo being supported, the sporophyte generation being supported by the gametophyte generation. We don't call these sporophytes and gametophytes and charophytes because they're haploid dominant and they don't really have an alternation between multicellular haploid and diploid. It's haploid dominant and just one diploid cell. It's not a multicellular organism, so we don't actually consider that to be the same as an alternation of well, it's not the same as an alternation of generations. So this haploid generation being supported by the diploid zygote, that's another pre-adaptation, and actually these are sometimes called pre-embryophytes for that reason. All right, cool. Okay, so I'm gonna stop right there, and next time we'll move into looking at. Actually, no, I'm not going to stop there. I'm going to go through one more slide here because we've looked at this already. And so now that we've actually seen some of these characteristics, I want to show you this again. This is just another timeline for the fossil record of, for land plants. And this starts with the Ordovician, which is about 450 million years ago. And what we see is, remember, the spore tetrads are our first evidence for land plants. And so spores being the product of meiosis, we see those 4 spore tetrads, and those are that first thing that we see in the fossil record. We also see plant cuticles arising. So we've got the cuticle, which is our indication for another land plant synapomorphy. And then the last one that I want to mention here is these things that we call plant tubes. I had this on our slide looking at mosses, but another name for these are the hydroids. And hydroids I had on the, let me just go back a couple slides here. If we look at this, we see hydroids evolving in mosses. And so I put on there are these are sort of prevascular tissue is what those are. So hydroids are prevascular tissue. They're not the same, as we'll learn in just a little bit what vascular tissue actually is. These are prevascular tissue that are found just in mosses. And this allows for, in mosses, having an upright gametophyte, which is going to be the next step towards getting a bigger plant body and evolving real vascular tissue. So this allows for the evolution of an upright gametophyte generation. Okay, I will stop here. When we come back, I'm going to look at, now that we've talked about several of these groups, we have our, we've set up our phylogeny with having bryophytes being paraphyletic, sister to the vascular plants. We can now start to look at how the different life cycles and the different stages of the life cycle have evolved from our green algal ancestors through all of our land plant lineages. Okay, stay tuned for more. Thanks.